Welcome, everybody. My name is Shobha Subramanian, and I'm very excited to invite you to the third annual Postdoc 180 event. Postdoc 180 is an event that is modeled after the 3-minute thesis from the University of Queensland in Australia. And we have adapted this event for our postdoctoral scholars at the University of Michigan Medical School. Postdoc 180 every year has been a very exciting day and an exciting weeks leading into that event. And it's particularly exciting in 2020 that we have managed to do this event over Zoom. And I really want to thank all of the Postdoc 180 participants who have been brave and have taken time away from their busy research schedules in the pandemic year to prepare and practice for this amazing 3-minute speech that you will be hearing from each of them. I also want to take a moment to thank my colleagues, Maggie Gardner and Beth Bodia, both of whom have been phenomenal and instrumental in putting together this event. So for many of you here who are not in the medical school or who are not in academia, I saw some family members earlier. You may be wondering what a postdoc is. Postdoc is the transition time between their PhD, when they have completed the PhD and before they go on to the job market. And this is the time when they hone skills to be more independent, both in research and in their skills, moving toward an independent career. And this is a cartoon that I'll let you read that we picked from the PhD comics, a famous cartoonist who does this, Jorge Sham, about the life of a postdoc. So, like I said, postdocs do a lot of research and contribute tremendously to the research enterprise at Michigan Medicine, and postdocs all over the world do this. And for example, the vaccine that, the first person in Britain that got the COVID vaccine, a lot of that is owing to basic research and clinical research that are done by postdoctoral scholars. Postdocs also write and communicate their research, both to a lay and a technical audience. And they collaborate across disciplines and mentor students and postdocs along the way in disseminating their knowledge. So postdocs contribute tremendously to the research in any university, especially at the University of Michigan Medical School, where we have nearly 600 postdoctoral scholars among us. So where do we fit in? So my team in the Office of Graduate and Postdoctoral Studies supports our postdoctoral scholars in their career and professional development journey. And you can imagine that this starts with career awareness. We help them assess skills. We help our postdocs build knowledge. We help them communicate their research. We help them with portfolio building, engage in other types of professional development activities, do some experiential learning. And ultimately, our goal is to have them a productive job search so they can land in the career of their choice. And Maggie and Beth are both instrumental members of this team. The Postdoc 180 competition itself kind of takes a piece of that particular map that I showed you and helps our postdocs hone communication skills, build confidence. It's an opportunity for the postdocs to showcase and share all the cool research that they do to a broad audience. It gives them a chance to engage with the broader community of fellow postdocs, faculty, and other professionals within the medical school and beyond. And today they'll also be competing for some professional development funds. And in today's competition, what's going to happen is that we have 3 judges who I'll introduce in a moment. And the judges will be ranking our postdocs on a few criteria, which I'll share again in a couple of slides. And the postdocs will be receiving award toward their own professional development. So we will give them funds based on some professional activities that they want to engage in that requires some dollar. And in this case, we are going to give 3 awards to 3 different postdocs, but it will be unranked, and each of the awards will be to the order of $150 each. We'll also have a People's Choice Award that you as an audience will get to vote. And when it comes to that moment, Beth, my colleague, will give you instructions on how to cast your vote. So I wanted to take this moment to thank 2 of my colleagues, Lisa Gubachi, our Administrative Director, and Mary O'Riordan, who's our Associate Dean for Graduate and Postdoctoral Studies. They really advocated in this unusual year of budget freezes and budget cuts to keep some of the funds available so we can do this event and use some of our funding for the professional development of our postdocs who are going to participate in this particular program. And this was not an easy task. So a huge thank you to Lisa and Mary for advocating on behalf of our postdocs. So now comes the fun part where I get to introduce the judges and I'll give you the rubric so you know what the judges will be scoring our participants on. So first of all, I want to thank Molly, Bryce, and Paula, our three judges who have taken time to volunteer for this service during the December of 2020, not an easy task. So a huge thank you from the bottom of my heart. Molly Kleinman, who is the program manager for the STPP, or the Science, Technology, and Public Policy program at the Ford School of Public Policy, is really well aware of why policy, science policy is important. And she has a pretty critical lens on policy making and using non-technical jargon in convincing a large set of audience about why we should move ahead with science funding and such. Bryce Pills, who is the Director of Licensing at the Office of Tech Transfer, sees a variety of patent applications and other types of technology coming through the University of Michigan researchers across disciplines, and has a pretty good lens as to how communication is something that needs to be critical to convince an audience who may be getting to decide whether something is patent worthy or not, as a simple example. And Paula Ross, who's the Administrative Director for the RISE program, Research, Innovation, Scholarship and Education program. And she comes to us from Michigan Medicine, and she has a very keen eye on many aspects of innovation, education, and research. And she kind of is at the confluence of all of this, especially in the health sciences, and has deep knowledge of working with the diverse set of stakeholders at the clinical and basic science fields. And she brings in that expertise of innovation and scholarship. So I really want to thank once again, Molly, Bryce, and Paula. And what the three of them have in front of them is this particular rubric. They are going to be judging our postdocs on three main criteria: comprehension, engagement, and communication. For comprehension, they're going to really think about, was the background sufficient, and whether the speaker had a logical flow to how they were communicating their science. In terms of engagement, they're going to think about if they captured the audience's attention, if they were enthusiastic, and if they made the audience want to kind of know more, like if they made them more curious about that particular area of research. And then finally, they're going to think about their communication skills in terms of clarity and how concise the presentation was, and if the content and delivery was appropriate for a wide and educated but lay audience. So this is the rubric, and they're going to be scoring each of the participants, and at the end, they'll come to the conclusion among the three of them who the top three are unranked. So, oops, I'm going to give it over to Maggie, who will share her slides because she has all the latest slides that our postdocs have given, and we'll go from there. And Maggie, if you don't mind, kind of reminding the postdocs of the time, that would be great.
Yes, absolutely. Thank you, Shobha. All right, so welcome to the presentation portion of Postdoc 180. It is my pleasure to introduce each one of our participants. I will be announcing them in alphabetical order. I'll ask each one to unmute and turn on their video, and I will highlight them so they will be front and center for all of you. They will have 3 minutes to deliver their speech, and then we will give the judges a couple of minutes to mark their scorecards. So it is my pleasure to introduce our first speaker, Dr. Anoop Arunagiri. Dr. Arunagiri is a postdoc in Peter Arvan's lab in Metabolism, Endocrinology, and Diabetes. He is presenting today his speech titled Protein Folding Problems in Diabetes. Anoop, are you ready?
Yeah, so I don't see the timer yet.
You see it
Oh,
now?
it's hidden there. Okay. Can you move it to a little left, the timer? Little bit of left.
Sure thing.
Yeah, thanks.
Good.
Thank you. Yeah.
All right. Take it away.
Okay. Millions of people around the world have diabetes. Family history, genetics, lifestyle, food, inactivity, weight gain, all of these have a role to play. Yet there's no definite cure for this. We in Arvan lab at UFM are trying to understand the causes behind diabetes, and we look at diabetes from a protein folding perspective. I'll come to what is protein folding in a minute. Right after we have our meals, our blood glucose levels go up. Then the body says to itself, hey, make more insulin. Insulin is the master hormone that controls the blood glucose. But have you ever wondered what makes insulin in the first place? It's this parent protein called proinsulin that makes insulin. We study proinsulin and its folding in diabetes. So, what is this folding? All of the proteins made inside our body have a specific job to do. And to do that, they have to first fold and take up a unique shape. Likewise, proinsulin has to take up this shape so that it can do its job of making insulin. Now let's take a quick look at proinsulin shape in a healthy condition versus a diabetic one. In the healthy one, you see these paper clips. These are like good folded proinsulins. They are all in shape, so they can go forward, make insulin, happy, healthy. Now in the diabetic one, you see these paper clips. These are clearly not in shape. We call this bad folding or misfolding. These misfolded ones, they are the bad guys. What do? They connect with each other and make chains like this. Not only that, just like in the heist movies like Inside Man or Die Hard, where a bunch of bad guys take control over good people and affect the normal functioning of banks or buildings, the very same way, these bad proinsulins take complete control over the good proinsulins and further makes chains and large bundles like this, preventing the good proinsulins from making insulin. That's a bad news, right? Because less and less insulin made means more blood glucose, diabetes, stroke, and other problems. So as you can see, this protein folding problem is an important cause to diabetes. Having said all that, I have two good things to share with you. The first thing is that we have now come up with a method that can actually visualize these chains of bad proinsulins in diabetes. Thereby, we can distinguish a diabetic from non-diabetic. The second interesting thing is that the majority of these chains of bad proinsulins are made in the early stages in diabetes, which means if we come up with strategies to fix this folding problem in these early stages, we are more likely to prevent diabetes. So it looks like just not the shape of you, but also the shape of the protein made inside you can be a predictor of diabetes. Thank you.
Thank you, Anoop. Let's all give Anoop a round of applause. All right, so we are going to give the judges just a few minutes to mark their evaluations. And I am going to, in the meantime, pull up another one of our favorite PhD comics while we wait. I thought this particular PhD comic was especially relevant given that we are coming up on our holiday days. And some of us might be thinking about taking a little bit of a staycation or resting and relaxing. I hope everyone is doing something to take care of their well-being. Okay, I'm going to move on to our next speaker. Next up, we have Dr. Serbi Galat. Dr. Galat is a postdoc in Malcolm Lowe's lab in the Department of Molecular and Integrative Physiology, and she will be presenting her talk titled Lose Weight, No Diet, No Exercise, No Pills. Dr. Galat, will you please turn on your microphone for us?
I'm unmuted. Can you hear me?
Yes, you are all set. Take it away.
So let me know when I have to start.
Go ahead.
Obesity is a very common and serious disease worldwide. How common is this? On an average, 1 out of every 3 adults are obese, which is about 35% of the population. Yes, it follows the same pattern as votes for Trump in mail-in ballots, 1 in 3. And this does not stop here. Obese people tend to develop other serious diseases like diabetes, heart disease, and some types of cancers. An obese person's risk of heart attack is 3 times greater than a person with healthy weight. So it is very urgent that we learn more about what might make a person obese. We all know that obesity is being overweight. We know that one can lose weight by eating sensibly and exercising. But these often come at an expense of increasingly unsustainable methods. And you can see some of the pictures in the middle of the slide showing just exactly that. Well, jokes apart. While that is true in a way, have you ever thought of losing weight just by making changes in brain? This is based on the idea that there is a connection between body weight and brain health, and our brain can fight against obesity. Yes, your brain controls your weight. Okay, let's think this way. Brain is the central computer that keeps body in check. Brain works through a set of neurons like how a computer works through a network. Just like we can alter code to get desired outputs, we ask the question, can we manipulate the brain cells to influence brain health and hence obesity? So, what I'm trying to do is to find the important genes in the brain's battle against obesity. How do we do that? Can we make changes directly in the human brain? No. We use animal models to identify these genes by increasing or decreasing their numbers in the brain and studying behavior. In doing so, we have found one master player in the brain, the POMC gene, that can fight against obesity. Next, we asked, can we increase this gene to reduce obesity? Yes. Simply by increasing the POMC gene in the brain, we saw body weight and fat loss. As a new strategy, we tried taking the help of important soldiers in the brain as well, the stem cells. These cells have this unique ability to make changes or reboot the brain. We used them to increase the POMC numbers and found the positive effects in terms of body weight and fat loss. Now you all must be wondering that is it really going to work? Then I would say yes. There are drugs out there targeting POMC gene and are under critical trials for the treatment of obesity. So there is hope. And I'm hopeful that our findings will lead to new treatments for obesity and will work for the treatment of for the betterment of humanity. Thanks.
Thank you, Dr. Galat. Everyone give Dr. Galat a hand. A round of applause. All right. Let's give our judges a couple of minutes to mark their scorecards. And I'm going to take us to our next PhD comic. All right. I'll remind the audience, please feel free to use the chat to cheer on and congratulate our speakers as they go. I know it's a little bit difficult to applaud and cheer people on. So we do what we can, but please feel free to use that chat. All right, I'm going to introduce our next speaker. I am pleased to introduce Dr. Ramkumar Mohan. He is a postdoc in pharmacology in the lab of Dr. Arun Anaratham. And his speech is titled, Watch and Learn to Prevent Diabetes, How Synaptotagments Control the Release of Insulin. Ramkumar, Dr. Mohan, would you mind turning on your video and unmuting?
Can you hear me?
Yes.
That's good.
All right. I'm going to move the timer for you. And you are good to go.
I'm ready.
Go ahead.
Hello, everyone. Thank you for your interest in my research. It's highly unlikely that you haven't heard about insulin. We all must have seen at least one of our family members or friends taking a daily shot of insulin. My research is aimed at improving their lives. When we eat, like our blood glucose level, commonly called the sugar level, increases. Insulin is the only hormone in your body that helps to bring down the elevated sugar level to a normal range. Failure to do so will result in high glucose, and if kept high over a prolonged period, this causes diabetes. So if you have a good insulin secretion, then you're young and healthy and happy. But then if you don't have a good insulin secretion, then you are diabetic. So there are specialized cells in your body that produces this precious hormone. And inside every single one of them, you have about thousands of insulin molecules being made and stored, but only a fraction of it gets released. So one might wonder, with so much insulin available inside your own body, why would anyone end up needing insulin from outside? Let me simplify this. Insulin is like having money in your bank account. Imagine you have all the money that you need for lifetime, but cannot access it. Well, that's how it's here. To solve this, we must understand what controls insulin secretion. This is where synaptotagments come into the play. Synaptotagments are a family of proteins that act like both sensor and also a key in your body that controls the insulin secretion. It detects the increase in glucose level by sensing the elevated calcium level in your cell, and then opens the door for insulin to be released out to bring down the elevated sugar level. We believe that when these sensors malfunction, insulin release is no longer in synchrony. So we predict that synaptotagments are the ones that decide when and how much insulin should be released. To test this, we developed novel genetic tools using mouse as an animal model. With that, we are able to actually see every single insulin molecule that are being secreted from live mouse cells. How did we do that? We made insulin to glow green inside the mouse body without affecting anything else. We call these mice Grinch mice, and we further took these mice and genetically removed synaptotagments from insulin-producing cells to further understand how insulin secretion is affected in these mice. So this brings me down to the title of my talk. I say we watch the release of these green fluorescent insulin molecules and learn the role of synaptotagments specifically and use this knowledge to prevent diabetes. We are currently expanding the scope of the imaging platform to human cells so that the translational scope of studies can be improved. So overall, I would like to conclude by saying that my project has a great potential in increasing or improving the current treatment options that are available for diabetic patients. Thank you, and stay safe.
Wonderful. Thank you, Dr. Mohan. Round of applause. All right, judges, mark your scorecards. I don't know about any of you, but during grad school, this refrigerator pictured here is all too familiar. Or maybe those of you who had roommates in college or grad school. Okay. Judges, let's move along. Our next speaker is going to be Alejandra Modina. She's a doctor of veterinary medicine and a postdoc in the anesthesiology department with Dr. Giancarlo Vanini. Dr. Modina, do you mind turning on your microphone and your video?
Yes, I'm here. Thank you so much for the presentation.
Awesome. So Dr. Modina's presentation today is titled Glutamatergic Neurons in the Preoptic Hypothalamus Promote Wakefulness, Destabilize NREM Sleep, and Suppress REM Sleep. Whenever you're ready.
Thank you so much. Well, 2020 has been a very hard year with the COVID pandemic and the lockdowns. However, believe it or not, pandemics have some good points. For instance, they have helped us to understand sleep. Between 1917 and 1930, a pandemic of encephalitis lethargica spread around the world, affecting 5 million people. Dr. Konstantin von Economo studied the birth of those and he discovered an area that makes us sleep. Since then, several clinical and experimental studies have confirmed that there's a specific area in the brain that is critical for sleep generation. This is called the preoptic area of the hypothalamus. However, this year, casually also during a pandemic, we discovered that a specific subset of neurons within this area does exactly the opposite. It promotes wakefulness and disrupts sleep. I know you might be wondering, how can this be possible? So here's the answer. In the past, scientists didn't have the tools that we have nowadays. They based their studies in anatomical areas, and it was really hard to determine if they were specific groups of neurons with specific roles. For this study, we use an advanced technique called chemogenetics. With this tool, we can activate in mice a specific subset of neuron in a specific area. We can select the neurons that we want to study by means of the molecules that they produce. In this case, we selected the glutamatergic neurons. They produce glutamate, that is the most common excitatory molecule that the neurons use to communicate. So we activated these neurons and then we recorded the electrical activity of the brain during 6 hours to determine how much time the mice spent in wakefulness and how much in sleep. And as I told you before, we demonstrated that the activation of these neurons increases wakefulness and decreases sleep. Also, sleep was highly fragmented. Mice were constantly waking up from sleep. Finally, a specific phase of sleep, that is where most of the dreams happen, was completely abolished. So, to sum up, we have demonstrated that the preoptic area of the hypothalamus is a much more diverse area than what we thought before, with some neurons that, yes, promote sleep, but some that promote wakefulness. We believe that this has important translational implications because these neurons may be involved in the etiology of the disrupted sleep that we see in some diseases, such as dementia, or some sleep disorders, like sleep apnea. Thank you so much for your attention.
Round of applause, Dr. Modina. All right. We'll give the judges just a couple minutes to mark their scores. I don't know about all of you, but I always appreciate good science jokes. If anybody has some good science jokes and would like to share them in the chat, please go ahead. All right, we've got some good jokes flying in the chat. I'm going to call our judges back and move on to our next speaker, though. Our next speaker is Dr. Matthew Ostrowski. Dr. Ostrowski, would you mind turning on your video and microphone?
You hear me? See me?
There you are. Dr. Ostrowski is a postdoc in Eric Martin's lab in the Department of Microbiology and Immunology. His speech is titled Food Additives and the Microbiome.
Hi,
All right,
everyone.
take it away.
So in general, we think about bacteria as being bad and responsible for causing disease. But in addition to bacteria that cause disease, there are hundreds of different species of bacteria that live inside our bodies and help us live healthy and happy lives. These bacteria help us digest our food, make vitamins that we need, and help train our immune systems. Another way these microbes keep us healthy is by preventing the bad microbes from taking over and causing disease. Especially after antibiotics disrupt the healthy microbiome in the gut, a pathogen called C. difficile can set up shop and cause life-threatening disease. Although drugs against C. difficile work to some extent, a treatment that has worked exceptionally well has been to supplement the patient's microbiome with bacteria from a healthy person. Although this clearly shows that the microbes create diseases, there's still a lot we don't know about how the microbiome works. One of the biggest factors affecting which microbes live in our bodies and what they're doing is our diet. Humans have been eating different plants and animals for thousands of years, but in the last century, we've started using various food additives to improve the texture, taste, and shelf life of food. One of these food additives is xanthan gum, which you can find in all sorts of food like salad dressing, ice cream, and low-fat foods. Although it's a food additive, xanthan gum is actually a material produced by a microbe that causes black rot on some common vegetable plants like broccoli and Brussels sprouts. So what's this xanthan gum from a plant microbe doing in our food? Well, it turns out it's a really good thickener and stabilizer and can improve the texture of a lot of foods. Many of you may also have heard of celiac disease, which prevents people from consuming food that contains gluten, which is a large component of wheat products like bread. Baked goods that rely on wheat for their texture can be made using xanthan gum, so people with celiac disease can still have gluten-free baked goods that mimic those made with wheat. Although it's been used safely for decades, my research has focused on how the microbes in our gut are affected by xanthan gum. So far, we've found 2 microbes that eat xanthan gum that seem to be in humans consuming a modern diet, but not people who consume a less industrialized diet. Although xanthan gum is generally recognized as a safe food additive and can help celiac patients eat gluten-free baked goods, it's clearly affecting which bacteria are living in our microbiomes. And with growing evidence that the bacteria inside us affect our health, I'm excited about understanding how food additives like xanthan gum are influencing our microbiomes. Thanks for listening.
Excellent. Thank you, Dr. Ostrowski. A big round of applause for you. All right, judges, we'll give you a couple of minutes. With this particular comic, I will put a shameless plug in for those of you who are graduate students and postdocs in the medical school. The Office of Graduate and Postdoctoral Studies Career and Professional Development team does do CV and resume workshops and individual advising. So if you are interested in something that's maybe a little bit more helpful than what's depicted in this comic, please feel free to reach out to me. And we'll talk about how to convert your CV into a resume. All right, moving on. I would like to welcome to the proverbial stage Jitendra Pant. Dr. Pant is a postdoc in biomedical engineering in Brendan Baker's lab. Dr. Pant, would you mind turning on your microphone and video?
Yeah, am I audible and visible?
Yes, you are. Thank you very much.
All right.
All right, Dr. Pant's speech today is titled Overcoming the Diabetic Epidemic with Beta Cell Implant. Ready when you are.
Diabetes is the 7th leading cause of death worldwide. Today, we are looking at 350 million cases of diabetes globally. This is higher than the current population of the United States. Every year, we spend $760 billion towards the treatment of diabetic patients. $760 billion. When you think about it, this is higher than the standalone GDP of 175 countries. So what is this big deal about type 1 diabetes? Type 1 diabetes is a disorder in the human body in which insulin-producing beta cells in the pancreas are mistakenly attacked by human immune system. And there is no cure for type 1 diabetes. And as a result, people with type 1 diabetes have to rely on insulin injection for the rest of their life. And when you think of pancreatic transplant, it is limited by donor availability because there is a waiting time of 3 years. And this is in the US alone. When you think of rest of the world with 135 developing countries, most of the people cannot even imagine in their dream to afford a transplant worth $20,000. We are clearly in the middle of a diabetic epidemic. And when you think about it practically, people with type 1 diabetes are 3 times more prone to the risk of death as compared to the rest of the people without type 1 diabetes with COVID-19 infection. This is very alarming. But am I surprised? Not really. My mother and my uncles have diabetes. My grandmother and my uncle lost their lives to diabetes. One of my very close friends have diabetes. And statistics suggest that 1 in 3 Americans will have diabetes in the next 25 years. This is alarming and we need alternative. And that's where my research comes in picture. I am engineering implants that contains beta cells that release insulin just like human body does. And to make them functional, my implants contain microenvironment using the hydrogels that can facilitate the formation of blood vessels. Now these blood vessels can supply these beta cells with oxygen and nutrients to make them functional. What does this mean to you? This means that no more 3 years wait for insulin transplant. This means no more lifelong insulin therapies. And this also means cheaper treatment options that most of the people in the world can afford. This makes me very hopeful. And I look forward to the day when my technology will be saving millions of lives. And I'm very excited to bring this technology to you. Are you excited about it? Namaste.
Thank you, Dr. Pant. Big round of applause for you. All right, let's give the judges a couple of minutes. Okay. Our next presenter is Dr. Helen Rich. Dr. Rich, would you mind unmuting and turning on your video? Dr. Rich is a postdoc in Beth Moore's lab in the Department of Microbiology and Immunology. Their title is Balancing Immunity, Bacterial Infection During Influenza.
All right, great. Can you hear me?
Yes. Let me move the timer for
Thank
you.
you.
Go ahead.
All right. How long can you hold your breath? We breathe every second of every day without even thinking about it. Think about the last time it was difficult for you to breathe. You might have had a cold or strep throat, or maybe even something more severe. It's scary, right? Our lungs keep us alive. Since March, we've all had a stark reminder of what respiratory viruses can do to us. My research focuses on one of these, influenza, or the flu. Tens of millions of Americans get influenza every year, even though we have vaccines. Since this virus mutates so readily and rapidly, we have to make new vaccines to fight it each year. And those tens of millions of infections still cause tens of thousands of deaths. One big killer during influenza is bacterial infection. Normally, bacteria live in our noses, happily coexisting with us just fine. But when we get the flu, which damages our lungs, those bacteria go down our nasal passages into our lungs and go set up shop in those little niches and nooks and crannies that those cells used to live in. And they eat up all the nutrients of those dead cells, and they proliferate with abandon. We have immune cells, white blood cells, like in the childhood picture, right to my either right or left. They come in from the bloodstream, those white blood cells. You can think of them as soldiers, which is good to fight the infection, but they bring a lot of fluid with them, which is very bad for our breathing. Lots of fluid in the lungs makes it very difficult to breathe. So my research focuses on a sensor, a bacterial sensor on these white blood cell soldiers called TLR9. TLR9 is a bacterial sensor. So you would think that if a cell lacks a bacterial sensor, it wouldn't be able to eat and kill bacteria. You can think of the way that these bacteria are killed inside these cells as these cells eating and digesting them inside little stomachs. However, when these cells lack TLR9, they're actually better able to eat and digest these bacteria. This was really surprising to us. And so we're trying to figure out, first of all, how we can balance immunity with not too many of these soldier cells coming in to fight the infection, while still having enough to be able to clear the bacteria. And as well, how we can figure out how bacterial sensors like TLR9 work in the context of bacterial infection during the flu, so that we can help people who get this disease, which happens every single year. Thank you so much.
Wonderful. Thank you, Dr. Rich. Round of applause. All right, judges, I'll give you a minute. All right. I would like to introduce our next speaker, Nadia Saadat. Dr. Saadat is a postdoc in pediatrics in Dr. Vasantha Padmanabhan's lab. Her presentation is titled Biosocial Impact on Black Births. Dr. Saadat, would you mind turning on your video and microphone?
Yeah, can you hear me?
Yes, I can.
Thank you. Just by looking at the picture of a bib as the study's logo, many of you will already agree to the importance of this study. Many times, we don't focus on all the factors influencing the health of the mothers and their babies. Compared to other races, African American women have more pregnancy complications and have the highest maternal death rate. Their babies have the highest number of infant deaths during the first year of their lives. These women are more likely to experience stress, depressive symptoms, and report discrimination. In this project, our goal is to study social and biological factors together to understand the mechanisms by which they contribute towards health problems for these mothers and their babies. Mothers' health and well-being is the health of future generations. According to the developmental origins of health and disease theory, the environment during pregnancy affects the health and disease status of children as babies and also as adults later in life. Inflammation is a one response how body fights with infection and injury. Inflammation is also found to be related to stress levels, depressive symptoms, obesity, and long-term diseases. This inflammation during pregnancy not only causes pregnancy complications for the mother, it can lead to long-term problems with immune system and other diseases for the baby. We have around 500 pregnant African-American women participating in our study from Metro Detroit and Columbus, Ohio. We have observed some association of plasma inflammation markers with depression, employment status, intimate partner violence, tobacco marijuana use, along with some long-term diseases like asthma and hypertension. Now we are studying the effects on fetal growth. By identifying these key factors and assessment of depressive symptoms, we can help reduce pregnancy complications among African-American mothers and the risk of long-term diseases in their babies. One of the approach we are applying is to recruit father of the baby to understand father's role on mother and baby's health in a sister study. Our research team is also developing lifestyle changes and community-based intervention strategies for the future research. Newborn health screening save lives. And by applying this kind of approach, we can contribute to save more lives and prevent some of the diseases to develop by focusing on improving mental and physical health of African-American mothers. Thank you.
Thank you, Dr. Saadat. Big round of applause. All right, judges, we'll give you about a minute, and then we will be moving on to our last speaker. I can't believe it, the time has flown by. Okay, I will be calling to the stage our last speaker, Dr. Isha Verma. Dr. Verma is a postdoc in neurology in Jacques Perrant's lab. Her title is Using Stem Cells to Study Brain Diseases. Dr. Verma, would you mind turning on your microphone and camera?
Yeah, can you hear me, Maggie?
Yes, you are all set. Take it away.
Thank you. Hi, today I'm going to talk about a brain disease called epilepsy. About 1 in 26 people in the United States develop epilepsy at some point in their life. So, what is epilepsy? Consider that our brain is like an electric circuit. In epilepsy, there's a problem in the electric circuit of our brain, which results in a loss of consciousness and involuntary body movements. There can be many causes of epilepsy, such as changes in our genetic material, trauma, or injury to the head. However, how these different agents can cause epilepsy is not clearly understood. One of the main reasons for this is a shortage of brain sample from epilepsy patients to study this disease. We cannot simply cut open a patient's brain to get a sample. Therefore, we need a different method to study this disease. And that's where my research comes into the picture. I work with a special type of cells called stem cells. Stem cells are like the body's raw material that can produce all the different cell types found in the human body. Consider them like the key food ingredients that can be combined to cook a variety of delicious dishes. So, where do we get these stem cells from? When we hear about stem cells, we might think that they're actually coming from human embryos, but that's not actually true. In fact, scientists have developed a new method to form stem cells without the use of a human embryo. For this, scientists generally collect skin cells or blood cells. A reset agent is added to these cells that changes these cells into stem cells. These stem cells can now be converted into different cell types. Throughout this whole process, the genetic material of the cells remains the same. In addition, scientists have also developed methods to convert stem cells into 3D-like mass of cells called organoids, which are like miniaturized and simplified versions of actual human organs. So, for my research, I collect skin cell or blood cells from the patients who are diagnosed with epilepsy. I convert these cells into stem cells and then redirect these stem cells to form brain cells and brain organoids. I ran a bunch of tests on these brain cells and brain organoids to understand the causes of epilepsy. We can also use this system to test different epilepsy drugs available in the market to check which drug will work best for the patient. Ultimately, this can help to develop personalized treatment therapy for the patient. This is very similar to the personalized diet regime or the skincare routine that some people use. And considering the high occurrence rate of epilepsy, that is 1 in 26 people, this can greatly improve the life quality of so many patients. Thank you.
Wonderful. Thank you, Dr. Verma. All right. Now, don't go anywhere. Our judges are going to go into a breakout room and deliberate over the top 3, but you have the opportunity now to vote for the People's Choice Award. I'm going to hand things over to my colleague, Beth Bodia, who is going to tell you how you can do that. Beth, go ahead.
Thank you, Maggie. Let me share my screen. Okay. There we go. Okay, cool. All right. So congratulations, everybody. That was amazing. So we would like to give you a chance to vote on our 9 presenters. And so the way to do that is to go to pollev.com/PIBS, or you can even use your phone and just text the word PIBS to the number 37607. We'll give you plenty of time to get there and to do your voting. I'm going to change slides so you can see what it will look like once you get to the voting screen, and that information will still be up at the top. So again, you can see pollev.com/PIBS, and then you will get the list of our 9 presenters. So take some time, think about, I mean, yeah, I'm glad I'm not voting because they were all phenomenal, but we do need to have one People's Choice winner. So take a moment. If it would be helpful too, I'll change the slide one more time and revisit what their slides look like. So if you're also remembering this presenter with this slide, we'll take a moment and let you do some voting. True, only one choice. So you only get to pick one. Good question. And yeah, if anyone's having technical difficulties getting there, please let us know. It's up at the top of the screen. Again, the directions on how to get there. Oh, thank you. Someone let me know that the poll is not looking active. So let me, good question. Yeah, it may not tell you if it's submitted. Let me take a look. I will, oh, no, it looks like we do have votes coming in. So yes, once you hit submit, it's coming in on our side. We're not showing the live results so that we can keep you all waiting. So okay, it looks like Lisa's saying it does say at the top of her screen, response is submitted. If you text it in, it usually just gives you an auto reply. Yeah, good questions, folks.
I'll be honest, I'm very excited about this. I'm watching the polls come in, and it is going to be a very close race.
Oh yeah, it's like slightly moving. I'm watching it too.
I'm so excited to see so many people voting for their People's Choice Awards. Like you, Beth, I'm very happy that I'm not participating in the votes. It'd be a very difficult choice.
And I'm sure difficult for our judges to pick a top 3.
So difficult. Okay, I'm gonna pop into the judging room and we'll be right back with their verdict.
Thank you, Maggie. All right, well, folks, continue voting. We have just a couple of other slides to share, some advertisements.
Oh, hey, Maggie.
Hi, how's it going?
Yeah, I think the voting is done.
Awesome.
So
what do we have?
I'll let one of the, I'll have the judges share that with you.
Oh, sure. So our top 3 were Ramkumar Mohan, Helen Rich, and Isha Verma.
Awesome. All right. One sec. Let me make sure I get these dragged into my new slide deck. You said Ramkumar, Isha, and Helen?
And Helen, yep.
Helen and Isha. I'm so glad I'm not voting.
I really appreciated that we were trying to pick a top 3 as opposed to ranking them. It made it a lot easier.
Yeah.
Well, good. We were pretty close to consensus when we started. So,
Maggie, do we have the People's Choice in?
Just one moment. Oops, wrong thing. It looked like it was just wrapping up. Can we close? I'm asking Beth if we can close voting. It looks like Ramkumar is in the lead for People's Choice, with Alejandra as a very close second. So I will put that one in that slide. Shobha, do you want to make the announcement?
Yep, should we leave this room? Thank you, everybody, again, if I don't get to say in person. Yeah, and I think that the top 3 probably was an easier vote than to kind of rank them because everybody was so good.
Yeah, they're all great. This is fun.
Yeah, this is really cool.
All right. So Maggie, do we have your permission to exit? Because you're kind of the master coordinator.
Yep, I'm leaving too. Okay.
Okay, all right.
Bye.
Okay, Maggie must be back. I have one other slide to show as our judges return to us. So again, Shobha had shared this link in the chat earlier, but if you're looking to see what our team does, you can visit our website. We have a number of resources on there. So even if you're not looking to attend a live workshop or advising session, there are just some static resources to help you along the way as well.
All right, thank you so much, Beth, for facilitating the voting. I am taking over screen share because the results are in. I believe Shobha is going to walk us through those results.
All right, thank you, Maggie. So once again, I want to thank everybody for attending on this Tuesday afternoon. Thank you also to Dr. Maggie Gardner and Beth Bodia. I can't thank them enough for making this event so smoothly running. Thanks also to Dr. Kleinman, Pills, and Dr. Ross for taking the time for judging, and everybody at OGPS for their constant support. So now for the big moment. Before the big moment and the drum roll, I have even bigger good news that all of our 9 participants each are going to get an award of $150 for professional development funds that they can use from our office. And we'll be emailing them how they can use that. So everybody, all 9 of them get this award because we thought they did an amazing job. And I'm so glad I was not a judge having to vote on the top 3 unranked. So congratulations to all of you for taking time and doing a remarkable job in practicing. And everybody was spot on at 3 minutes or under and delivered a beautiful speech. So a huge round of applause to everybody, virtual applause. So now, Maggie, if you don't mind moving to the next slide.
Drum roll. It's all 3 of them on the next slide.
So this is our top 3 unranked. There's no rank order. Our top 3 are Helen Rich, Isha Verma, and Ramkumar Mohan. So congratulations, Dr. Rich, Dr. Verma, and Dr. Mohan. All of you did an amazing job. Congratulations, and each of you will get an additional $150 from our office for your professional development. And again, like I said, we'll give you some examples of what you can use it for, but those are funds earmarked for you to use. So, and the judges said that it was easy that they didn't have to rank them because that would have been a very difficult job. So the unranked made their work a little bit easier, although even the top 3 out of the 9, I think, was a very difficult decision to arrive at. Okay, now the people's choice. What have the people said?
So I am actually going to stop my share because as we were discussing this, the last little bit of votes trickled in and the result did change just at that last minute.
Why don't you announce it, Maggie, then? since you're looking at it live. Yeah.
I am looking at this live, and I am pleased to announce that our People's Choice winner is Alejandra Modina. Congratulations, Dr. Modina.
Congratulations, excellent.
Should have put in a soundtrack of roarous applause, because that's what you would be receiving if we were live. Congratulations to everyone.
Everybody can, unmute if you will, and we can applause together for a couple of seconds to pretend that we are in an auditorium. So if you don't mind joining me. All right, that officially concludes our Postdoc 180 for 2020, a very memorable, exciting, fun event. And I hope you'll join us in 2021, hopefully in person for another Postdoc 180 that we will definitely be doing around the fall of the year. So September is when we typically do it. So hopefully we'll be back on schedule. Everybody will be back in person. So hopeful thinking, and everybody be safe. Wear your mask, happy holidays, and take some time to relax and have a restful break. Thank you.
Thanks, everyone. Congratulations.